now I will welcome you again to this webinar on environmental DNA. We are very happy that so many people decided to join us today. And for those of you that are not part of Biodiversa Plus or might not be sure what this is, I will give you a short introduction on that, and then I will introduce the speakers today, and we will begin with the presentations. So, Biodiversa Plus is the European Biodiversity Partnership supporting excellent research on biodiversity with an impact on policy and society. It is organized in different groups for working, and we are part of the Work Package 2 that wants to promote and support transnational biodiversity monitoring, and concretely aims at setting up a network of harmonized schemes to improve monitoring of biodiversity and ecosystem services across Europe. This is very wide, but a lot of work. We have many different tasks involved in different aspects of this. We have tasks working on priorities and indicators, on harmonization of protocols and databases, use of biodiversity monitoring data, and we are part of the task 2.3 on novel technologies and citizen science involvement. We are all of this work is linked with the biodiversity monitoring pilots. We here have the pleasure of having 2 representatives of this pilot, so you will get to know more about this, about 2 of them, because we have plenty shortly, and all of this fits into another task that aims at building a network of national biodiversity monitoring schemes. Just for you to get a little bit about us. More concretely, our task on novel technologies and approaches. This year we launched a survey of novel technologies monitoring across partners of Biodiversa Plus, where we asked partners if they were using the 5 novel technologies that you could see in the right part of the image, including environmental DNA. So we would want to know what was the deployment state, or if they did not deploy these novel technologies, how interested they would be in using them, how did they use it, so what target taxa were they studying with this, or what essential biodiversity variables, so for example, if they were targeting a species population or ecosystem traits, and also as well, what challenges and constraints were they facing in the use of these novel technologies. And the image in the right represents most of the issues that they refer to. And as you can see in blue, lack of capacity and training was one very often referred for the 5 novel technologies. So all of these results are part of a report that is available on the Biodiversa Plus webpage. And this is the reason why we are here today. We are launching this series of webinars. We will be having more of those, so please stay. We will let you know. Probably we will be having one of bioacoustic for January, but still to launch to say the date. And yes, this was just a short introduction of the webinar and the speakers. And the first presentation would be by Katrine Hansen Lemming, but she's having some trouble, I think, still to connect. So maybe we will begin with Julia Seeber. She has introduced herself. She's an expert on soil biodiversity in montane soils. She is working in EURAC Research with the government of Bolzano, but she's also involved in the University of Innsbruck, and she's of course, she's the pilot coordinator of the soil project in Biodiversa Plus. Her focal group are earthworms. Then we will have here as well Nina Prasil-Delaval from PatriNat, France. She is a marine ecologist expert on fish biodiversity in coastal rocky fish, and she was the previous coordinator of the EuRockFish pilot for Biodiversa Plus. She's still very involved. and she will let us know a little bit about this very interesting pilot on marine environments. And as for Katrine Hansen Lemming, she will be introducing herself when she's able to get, which I'm sure she will. And she is a biologist working for the government of Denmark. She is coordinating an eDNA monitoring project for that government. She's working in this government since 2016. So maybe we will change a little bit the agenda, but let us begin. And of course, we will leave some time for questions and debate in the end. So please don't hesitate to share experiences or any questions that you might have for the speakers. And this part will not be recorded, so feel free to ask anything and your privacy will be safeguarded. Thank you so much. And Julia, if you would like to begin. Thank you.
Okay, yeah, thank you again for having me here to talk a bit about the soil pilot. I will very shortly introduce the pilot, just to give you a bit of background information, but then of course focus on the main topic today, the use of eDNA in our work. So we have been collecting data now for 2 years with our partners, and we are currently, as I said before, in the process of identifying all the many invertebrates that we collected during these 2 years, and of course also analyzing soil parameters and most of all the eDNA. One of the aims of this pilot is to define the advantages and limitations of traditional methods and to compare them with molecular methods, because it will be an important part of our final report in 2 years, in which we will recommend a transnational monitoring scheme for soil biodiversity. And of course, this will include a recommendation on methods. So in the pilot, we have 10 partners from in and outside of Europe. We also have Israel and Turkey joining us. So we have a very good representation of the biogeographic regions. And I probably need to mention that we did our work in near natural forests. So for the fieldwork, we used the protocol of Soilborne. Some of you might know that is a global initiative aiming to understand soil biodiversity on a global scale. I won't go now into details on that protocol, but we use pitchfork traps to collect invertebrates living on the soil surface. We do hand sorting of soil cores to have the soil-dwelling invertebrates, and we also take bulk soil samples from each site to analyze soil characteristics and also, of course, the eDNA. So hopefully this way we will be able to compare results from the both types of methodologies. So we have the traditional methods where we pre-sort the samples, and then for some taxa we even do species identification with the help of an external company. And the second part is then the eDNA. We use the Lucas protocol here as the workflow we are using. Lucas is that big initiative analyzing soil parameters across Europe, but since 2018 they also do soil biodiversity. These are the primers we are using, just to give you a quick overview. We are targeting archaea, bacteria, fungi, and invertebrates. And for today, I will only focus on the results of the invertebrate data. So the big advantage of using eDNA would be that from one single sample, we would kind of more or less get the entire soil community. While if we target that via traditional methods, we would at least need 5 different methods to only target invertebrates. So I guess it's the way forward in the monitoring scheme to use eDNA for this. Just to show you some nice pictures from our fieldwork while we are pitfall trap in the middle, hand sorting of soil cores in the right picture, and also a soil sample. So now to the preliminary results of our eDNA. The first question we looked into was the storage of the samples, because best way would of course be to freeze the samples immediately in the field, but that's not always possible because it might be remote sites or you don't have the equipment or whatever. So we asked our partners to send us dried soil samples, but even though it's not yet statistically meaningful because the replicate number is not so high, we can already see that during the drying process, the alpha diversity of eukaryota really decreases. So that will be one question we need to answer for monitoring how to treat the samples before further processing them. When we look a bit more closely into the results of the first season of eDNA we got, we see that 40% of the sequences belong to Annelida, that would be earthworms and the like. And as might be expected, it should be earthworms because that is what we see as invertebrates in our samples, but all the sequences belong to enchytrates, and enchytrates are the small relatives of the earthworms. Then we have a lot of nematodes, which is good, but only 19% of the sequences belong to arthropods, which is a really main taxon in soil. If we then zoom in a bit more, because nematodes are not targeted with our traditional methods, it's about half the sequences belonging to Annelida, but again, it's enchytrates and not earthworms. And then about the other half belongs to arthropods. The direct comparison of the two methods, it's still kind of difficult, and I have to give a huge thank you to one of my partners, Sam Lambrechts from Belgium. He really did a huge effort and a lot of work on trying to give us a visualization of the results of the two methods. So to the left, you see what we find as specimens, as individuals in the traditional methods. And on the right side, you see what we get as sequences from the eDNA. The comparisons is also still difficult because we use the sequences and compare them to a reference database. Actually, we use two different ones, PR2 and Silva, but the taxonomy of these two reference databases is not uniform. So that's why I wrote partly order level, because the taxonomic levels are a bit mixed up in the reference database. So that is one problem. The maybe second issue is that not all the invertebrates that we find as species in our traditional methods are yet included as species in the sequence databases. And just from that very first glimpse into our results, you can see that most individuals that we identify are insects, which is more or less obvious because we have a lot of insect larvae living in the soil. We have lots of beetles living there. We also have spiders. So Arachnida is not a huge part, but still they can be found also in the soil samples. And of course, Myriapoda, so diplopods and helopods. If we then look to the right side, most of the sequences were Arachnida, but I have to say again, it's not spiders, but mites. So we almost found no spider sequences in the eDNA. It's just mites. And also, and if we look at the insects, it's a very, very small part that we found as sequences, almost no beetles. The only taxon that might be a bit more comparable are the Myriapoda. So that seems to be something that we need to be working on still for the next 2 years while the soil pilot is still ongoing. So currently we have that possibility to select between two different kinds of methodologies, but we do know that traditional methods are very time consuming, and it's also difficult to have experts helping us with species identification. That could all be easily solved by using eDNA, but there we have the problem of certain primer bias. That's why we need to work on the question which primers to use in the future for such a monitoring. Might be the solution to use group-specific primers. We need to work on the optimization of protocols and workflows, and then we need to address the issue also of the reference database, how we can improve getting sequences identified so that we can have a better overview of our soil biodiversity. I think, or I am very sure, that eDNA is the way forward, and this should be the method used in the monitoring, but we're almost there, but not quite yet. So thank you for listening. And I'm, of course, happy to answer questions.
Thank you so much, Julia. We will now continue with Nina.
Thanks.
So, hello everyone. As Gloria introduced me, I'm Nina Prasil-Delaval and I work in France on the EuRockFish pilot study. And today I'll talk to you in more detail about the use of eDNA for reef fish monitoring. So first of all, a big shout out to our wonderful partners that are located all around Europe, which will have its importance later on in terms of legal issues and so on when you organize such a pilot. And that did a wonderful job in joining us. So the pilot started this year, so we had to do the fieldwork this year as well, which is also have some implication later on. And yeah, which will explain a lot of the time constraints that we faced, but we did it, and a huge thank you for all of them for their support. So I'd like to start with the object of study, which are reef fish in European coastal waters. I hope you can see my mouse, but here we are talking about demersal fish. So the fish that resides near the rocky bottoms, not the pelagic fish that swims freely in the water column. And we are studying these fish in temperate rocky reefs, which includes kelp forests, but also mesophotic reefs in temperate settings, even though the methods that we will be talking about today could be also used to monitor marine infrastructures. As you may all know, fish communities are sensitive to human threats. They are facing fishing pressures, habitat degradation, climate change, but also the arrival of invasive species. And in Europe, there are several European directives and regional sea conventions that demand that reef fish be monitored to evaluate their good ecological state. So you can, maybe you know of the Marine Strategic Framework Directive or the Barcelona Convention, among the different conventions that they are. So the problematic that we are facing around Europe is that long-term monitoring is missing in several European countries. Reef fish are well monitored in some places and poorly monitored in others, and even at the scale of one country. Further, the observational methods used are heterogeneous, and the European reef fish scientific experts are not well organized in a network, which means that the information travels slowly or not at all, and it's hard to harmonize our methods. That's why the general aim of this project was to develop and test homogenized protocol for two traditional methods, scuba diving and video, that have been used since the beginning almost of reef fish monitoring, and a novel complementary method, which is metabarcoding on eDNA, which we are focusing on today. So we aim to validate that methodological framework and produce a methodological guide for monitoring European reef fish under MSFD, regional sea conventions, marine protected areas, and so on. So as I just told you, we are using two visual methods, which allow us to measure indices related to abundance, biomass, and body size distributions, in addition to species diversity. But today we are focusing on eDNA. And so the question is, why use eDNA to monitor European reef fish when we already have visual methods? So the fact is that these visual methods are visual, which means that when visibility is not good, you can't detect the species. When the species are small hidden fish or large, vibrant and or shy, elusive fish species, then you won't see them and you won't detect them. We're using eDNA to bypass this issue and to have a more inclusive view of species diversity in our reefs. And that's why you're here today. eDNA is on the rise. The number of publications, the number of projects using eDNA is increasing rapidly. But however, there's a huge variety of protocols used. And to monitor reef fish with eDNA, you can use different types of volume, 1 liter, 30 liter. The porosity of the filter you use can change. The type of sampling can change. You can use different primers for eDNA, for DNA amplification. You can do different types of molecular analysis. You can do your bioinformatic analysis differently. And this is proving to be a real issue when we want to analyze data and compare our results in different areas which do not have the same ecology and so on. So we really need to ask ourselves, how do we use eDNA collectively in a project involving several institutions and countries? Because most of the partners, when we asked them if they already use eDNA, they did. And they did it in a way that was different from us and that was different for each partner. So we had to find a way to do that as a whole. And we also had the additional constraints I talked about. So the limited time to launch the project, but also limited time during the fieldwork, because we have to deploy all three methods. And for example, for the methods that involve just the sampling of water and then to filter it in the lab, we do not have time to do that when we already have three methods to deploy. We also have limited space on the boat, so we cannot store water samples of 30 liters each. And it must be compatible with upscaling at European level, which is our objective. So the protocol that is chosen must be easy, user-friendly, and reproducible. So that's why the method we selected collectively was to filtrate directly in the field, to use filtration capsules of 30 liters to capture a maximum of diversity and rare species. And we agreed that only one private lab will be in charge of supplying all the kits and performing the analysis. Because, as you can read, we bought more than 400 kits, and this takes time to analyze, and for a 2 years long project, we cannot wait for public lab to do it. It's more expensive this way, but it was our only viable solution. So as the coordinator of the pilot, we had to launch a call for tender. I won't go into the detail of the technical requirements. If you're interested, you can always contact us or ask questions afterwards. But I will just go quickly through the different steps. So we had to make that list of technical requirements. Then the European call for tender was launched, and each respondent was able to provide his own technical solution based on our requirements. And then we had to analyze all these offers to select the best suited respondent. So yeah, that took quite some time, but we did it, and we managed to end all this before the fieldwork started. And so we had all our sampling kits, and we were able to start in July the fieldwork. So that's the system in itself, just to give you a quick overview of it. So we bought this pump independently from the call for tender. Each country bought his. So it's a peristaltic pump with 2 heads that you can see here. And on each head, you can attach a filtration capsule with its tubing, and that will filter the water through the capsule. And then we also had to decide on the field because, and I forgot to say that beforehand, but there are no official protocol for sampling eDNA for reef fish monitoring. So there are a few questions that we wanted to address in these pilots, which are where and how should we sample, at which depths, and should we do it in a mobile way, for example, from a boat at the surface, or while diving and carrying the pump in all the small crevices and so on that you can find on reefs, or should we do that in a stationary way? Should the sampling protocol depend on environmental local variables, for example, the water movement, for example, the tides, or the water stratification? Because when the water at the surface and at the bottom is separated, for example, with a thermocline, so a huge difference in temperature, or a halocline, which is a huge difference in salinity. If you filter just at the surface above these clines, with no mixing of water, maybe you won't have the full picture because it depends on how eDNA spreads in the water. But also the impossibility to dive in some countries because in a professional setup, it might be legally very constraining. We chose to do it in a stationary way for all partners. But in France, we also acquired additional funding from other sources to try the other methods. So to be able to compare in one place how the effects of the way of sampling impacts our data at the end. So as we just did the fieldwork, you have no results to show you today, but the conclusion will come at the end of the project in our methodological guide, so stay tuned if you're interested. And at this stage, we envision 2 possibilities. It's either standardization or harmonization, and that would be through a decision tree that would allow the user to select the sampling strategy that best suits his needs depending on the environmental context. And yeah, to give you the first feedback from the partners after the fieldwork, the main feedbacks are that it was easy to deploy, that it did not require a lot of formation. It's quite quick. In 1-2 days at most, you're able to do it perfectly. And the main issues that we faced, but it's quite rare, are linked either to the pump, which sometimes did not start, but when you do it in the stationary way, you cannot detect that it did not start because it's underwater and you can't see it. And sometimes algae got stuck and impacted the volume filtered. So we note all this on a sheet that we send to the lab, and we will keep track of it later for the analysis. So overall, there were good feedback on the method and its deployment on the field. However, we had some issues with customs for countries outside of the European Union. That's maybe very time consuming. So for example, we had in 1 country, in 2 countries actually, huge problems for the kits to go from France into the country, but also to export the kits after the sampling from the sampling country to France. So most samples arrived at the lab for most partners, but we still have one that we should be able to resolve soon, but it takes more time. And we should have the results from the lab back in March 2025, so that will mark the official start of the analysis. And we have collectively 1 remaining open question. that we might discuss later on if you have any idea or solution or are facing the same issue. It's on which online global biodiversity repository should we share this data? Because as of now, most of them are not prepared to host presence data from such methods with all of the eDNA sequences. So yeah, thank you for your attention. And if you have any question, please do not hesitate.
Thank you so much, Nina, for this excellent presentation. Let's continue with Katrine.
Thank you, Gloria. I'm Katrine, and I'm working for the Agency for Green Transition and Aquatic Environment, a governmental agency in Denmark. And we have been working on eDNA in waterholes, focusing on great crested newt and the water beetle that I'm not going to try to pronounce the Latin name of, and another diver beetle. There you go. I'm a Master of Science in Biology, and I've been working in the agency since 2016, and I've been working mainly with the water courses and Natura 2000, and now with the eDNA in aquatic environments. So my agenda of the presentation is a more overall presentation of what we have been doing. And before that, I'd like to just make a short presentation of the Natura 2000 monitoring program, where we are looking into using the eDNA. Also a short introduction to what is eDNA. And this was meant to be in the beginning, but I'm very sorry. You'll have it now. And then I'll go through the aim and some of the aim and objectives of our eDNA project. And lastly, what have we learned, and show you a few of our results up to now. So what is the monitoring program, and why do we monitor? And I will just go very briefly through this, but according to the Habitat Directive, Denmark and other members of the EU is obliged to implement the necessary measures for ensuring or restoring a favorable conservation status for the habitat nature types and the species covered by the directive. And at the same time, we are obliged via the implementation of the Bird Protection Directive to take appropriate measures to secure the national population of birds covered by the directive. And Denmark has chosen to do this by systematically and recurring planning in the individual Natura 2000 areas. And in Denmark, we have 250 Natura 2000 sites. And we work in a cycle of 6 years. So first, we map and monitor the habitat types and the species. Then we data analysis and send our reports to the EU Commission. Then we start the planning the goals and the restoring plans for the areas, and then we do the actual action plans, and then the cycle repeats. But what we experience is that some of the conventional monitoring are time-consuming, and also looking into the new biodiversity strategy coming up, we have even more monitoring to do. So we were interested in learning about other methods that might help us to be more efficient and hopefully higher quality of our monitoring program. So shortly, we were looking into eDNA. Shortly, what is it? Which advantages and potential does it have? But also the pitfalls with eDNA. Firstly, I would like to outline what eDNA is. eDNA is an abbreviation for environmental DNA. So that means DNA remains from animals or plants or bacteria or whatever, which can be found in various media. It could be water, soil, or even air. The latter method is still it's still working on the method. And it's most secure to use in closed or semi-closed environments, to my knowledge. When a sample has been taken, the sample can be analyzed in various ways. And in our project, we have focused mainly on 2 methods. We focused on the qPCR, which is a single species test. It detects 1 species in the sample. And we also focused on the metabarcoding, which is a several species test. So within a defined group, such as vertebrates or invertebrates, you can detect the species within that group. So the advantages and potential of eDNA, when I'm talking into this, it's important to know that we're talking into a method that is complete and working, because there are still disadvantages. But we have a method and it's complete, then eDNA can increase the quality of our data. And it can give us an effective answer to which species live in a given location. It can give us valid data with a high detection rate and a small margin of error. And it may be easier to find the rare species and few individuals in an area. It may simplify the monitoring processes. With harmonization and unification of the monitoring program can be created with the eDNA. We can also be lucky enough to combine several programs in one, making it more time-consuming, less time-consuming. And it may be a tool we can use to identify new potential habitat faster. And it may also be able to generate a more fulfilled, for example, plan list in aquatic environments in a monitoring context, instead of using a lot of time finding them on our own. And maybe it can be less weather dependent, especially for some of our species it will. It can ease the pressure of a busy field program. So the eDNA monitoring can take place over a longer period of time than the conventional programs that we have now. And it may be resource saving, so more locations may be researched faster, and time and money can be reduced when the method is scaled up. So this all sounds very promising, but of course there are also pitfalls that we need to be aware of. When looking at the data, we have to bear in mind that there is a risk of false positive values and also false negative values. So for example, it may be transferred DNA from one waterhole to another waterhole on our equipment. So hygiene is very important when sampling. And one way to check our hygiene is to use the negative control samples every day we go out. It can also go wrong in the analysis, and it can create both false and positive false positives and false negatives. This can be because there aren't enough DNA in the sample, or simply the analysis go wrong, or maybe imprecise, or data is interpreted the wrong way because there is no threshold or no limit of detection. We might have passing or migrating individuals in the waterholes, and they may leave some DNA trace. But depending on how many there are, the concentration is very little, the concentration of the DNA. But then it's also important to think about what is it we want to know. Do we want to know if the species is in this waterhole alone, or are we more interested in the area that it's living in? So, and then of course, there might be dead individuals leaving DNA traces to the water. And it's very important not to disturb the sediments where they line to not to get the DNA up in the open water when we take the sample. But no matter what, it's important to know that false negatives are both in the eDNA as well as in the conventional monitoring. So it's not that one way is better than the other. Okay, I will go through what we did in our project just in overall terms and how we did it. So to take to the focus of the webinar today, I have only included some of the objectives of the project. our overall objective or overall aim was to find out if it was possible to apply eDNA to our monitoring program at this stage. And to figure this out, we searched for inspiration and networking, and we were interested in finding inspiration on how to use eDNA in the monitoring programs. And in general, we were seeking more inspiration instead of inventing our own procedures. And we were curious to know if there was any protocols already where, so where are we at this point in the EU? Are there any harmonization yet between the DNA methods used across the EU countries? So we looked towards England, who already has implemented eDNA and use it to when they look for the great crested newt. And we also looked towards Germany, where we knew that we could find experiences with eDNA in a perspective of the Water Frame Directive, and to understand the importance of consequence of introducing new methods into an existing monitoring program and a long timeline of data. We also wanted to examine different laboratories. Were there any accredited laboratories to perform eDNA analysis? And which method did they use? And are there any standards yet? And to test the credibility of the different laboratories, we wanted to compare eDNA results from 2 different labs. Then we wanted to look at eDNA compared to a monitoring program. So we both collected eDNA samples as well as did the monitoring practice at the same location. And lastly, we also wanted to know specifically towards the different species. We knew that eDNA is good for great crested newts, but how does it work when we put it to large scale use and several field workers has to go and do sampling? And also look into, does the eDNA already, does it work for the 2 beetle species that we are looking for? Because these 2 beetles are very difficult to find with our conventional method, but we are afraid that they don't leave enough DNA traces in the water because they are protected with a hard outer shell. And how did we do it? So in 2023, we reached out to England to arrange several meetings, and we talked to John Littlefair, who at that time worked at Queen Mary University of London and works with eDNA in air. We also visited an English laboratory, which are accredited and very specialized in eDNA. This is what they do. And they also worked with the English standardized eDNA method for the monitoring of great crested newt, which is also put into their legislation. We also talked with the EURAC DNA Center of Excellence. This is a virtual network that brings together expertise on the DNA-based tools for monitoring for the environment. And they consist of member organizations that use DNA-based tools for monitoring the environment and the biodiversity, ecosystem functions, and responses to the changes within these. And furthermore, we connected to Florian Lese from Duisburg-Essen University in Germany. And he works on how eDNA may be implemented into the current monitoring practice with focus on the Water Frame Directive. So we thought we could do some parallel perspectives here. When choosing the laboratories, we decided to work with the English lab that we knew had a lot of experience. And then we also found a Danish lab that didn't have neither standardized methods nor accreditation. So it was very interesting for us to see whether they got the same results. For the parallel testing, we collected samples for both laboratories, and at the same time, on the same locations, we carried out the conventional monitoring practice. And then we tested eDNA for great crested newt on 230 locations, and we had about 10 field workers that put the method to test. And we also tested the eDNA as a tool for the beetles, but only on 12 locations because we don't have many locations in Denmark with these beetles. So based on the results, we concluded on the applicability of eDNA on large scale with many field workers, as well as whether eDNA can be used as a monitoring tool for the beetles. And I will go through a little of the results later on. So what did we experience and which results do we have up till now? I can say that the project is not done yet, but we have learned a lot so far. So networking. I will just go through some of the points that we learned from our networks and our talks. And when we talked to Joanne Littlefair, we learned that eDNA can be found in different substrates and has a larger perspective than we thought it had. She had worked a lot with airborne eDNA and has put it to test, for example, with zoo animals, and also worked with it on bats. So our reflection upon this was that it sounded very interesting and promising, but for some habitat directive Annex 4 species, but also that eDNA still has its limits, and not all species are fit for eDNA yet. Then we spoke with Florian Lese, and we talked about the advantages and disadvantages with eDNA and how far the development of eDNA is. And some of the advantages he talked about was that eDNA, the method, is less disruptive to the environment, and we don't need to collect specimens. We can only collect a water sample or soil sample, etc. But some of the disadvantages is that there are no standards yet, and some parts of eDNA are not ready for the monitoring method. We also talked about that eDNA might be useful in some areas in the monitoring program, especially in relation to the Water Frame Directive, but within the Habitat Directive, there is no standardized method yet. And Florian advised us that there should be the standards, so it's possible to compare the results between countries. We also talked about whether eDNA can be used to find the quantity of the individuals, and he advised us to think more in groups, such as less abundant or more abundant than specific numbers. Sorry. So our reflection upon this was that we should be careful not to sort of become an eDNA front runner, but we need to be aware of how if we implement eDNA in the monitoring program, and the focus should be more on the laboratory's accreditation and standardized methods. Then we visited the Nature Metrics, the English laboratory, and we heard about different successful eDNA projects of theirs, as well as how it works with the great crested newt. And we talked about sampling in different medias, and we talked about the metabarcoding and its range. So we learned a lot about the eDNA, and based on their knowledge, we had most of our samples analyzed in their lab. When we talked to DEFRA, we learned that eDNA is a tool and not a full package or a full toolbox. eDNA cannot do the same as our conventional monitoring program, and therefore eDNA cannot be a substitute. eDNA can do something else, and the question is therefore how and where eDNA can add something positive to our monitoring. eDNA does not provide definite answers, but eDNA can be used as an early warning system, for example, signs of invasive species. And they encouraged us to collaborate across the EU countries and seek knowledge in existing eDNA networks so that countries across the EU use of eDNA is harmonized. So next is our results, some of our results from the project, but I will just go through some of them. So this doesn't take too long. But to show how the parallel testing worked, I would like to show you the qPCR results that we have. So here we see the results for great crested newt from the conventional monitoring, as well as the eDNA results from the two laboratories. And the results has to be compared to each other, and I'll just shortly go through this. So first, Of course, the interesting test would be to know if we find them with the conventional method, do we also find them with the eDNA method? And that's what this diagram shows. So to explain the diagram, you need to know a little more about the underlying data. And in order to be able to compare the findings one-to-one between the different laboratories, it is the exactly same locations that are shown in the diagram. So here, for example, we have compared the conventional method with the English lab on the same locations as the Danish lab. The Danish lab had fewer samples, but because the English lab got more, this is the sum of all the locations, because it's still interesting to see are they still agree with the conventional method. So what we see overall is a very good correlation between them all. The last two pairs shows the correlation between the two labs, and they have more or less the same results. The 3% is two locations where they disagree. And over here, 7, 2, 3, it's also very few locations. And it seems that there has been some challenges with the samples. So it could be, for example, if there is a lot of cattle in the areas that urinates into the water, it will affect the quality of the samples. We also have negative controls to keep an eye on our hygiene in the field. And the 5% here is also just two samples, that has a tiny trace of eDNA, of DNA in the sampling. And because of this, next year we will begin to talk about thresholds. So where from what concentration of eDNA in a sample will we accept that it is a positive because of the false positive possibility. The diagrams for the beetles are based on the same principles as the great crested newts, but the English laboratory did not offer qPCR analysis for the beetles, so this is only for one laboratory. If we look at the conventional window, that meaning when can we go to the field and do our monitoring with the conventional method, we go out in May and we go out in September. And we wanted to test, can we do it further than that? Can we go into April as well? So it seems that in May for both species, there is a rather good correlation, actually better than expected, because we didn't expect a lot of DNA traces in the water. And it seems that eDNA might also work in April for the diving beetle. But it needs to be said again that these results are based on very few locations, so therefore it requires further investigations to say anything final. If we look at the negative controls for both species, there are no contamination. Yes. So which potential challenges do we know of from our projects? And which have we thought of? So of course, the false positives, as mentioned, the false positives may be handled, we think may be handled with a threshold or a limit of detection, as well as good hygiene in the field. But we also need to remember that no monitoring method is flawless, so it's important to decide which is the better one. Another challenge that we can be how we convince others that eDNA works. Now that we don't see the individuals of the species, but we only see traces of DNA in the water. So we think it's very important to make this parallel testing between the conventional method and the eDNA method before changing a method, but also during a period. So from time to time, at least once during the 6 year of period, we go out and have an expert checkup with a conventional method. Another challenge might be how to argue for changing a monitoring method. So to avoid disturbing a timeline too much, it is important not to change the method within a period of monitoring, but from the beginning of a new one. Then we also need to look into other kinds of arguments. Why would we want to change a method? So if it's improving an existing method, it could be an argument for changing. Also, if it's working towards harmonization across the countries. Sorry. And then we have to remember that if we get at least as good results, we will probably get it cheaper in the long run when we talk about eDNA. And the method is less subjective than the current methods we are using. So a challenge is also to ensure that the results are correct when the results now are out of our hands or the field workers' hands, and instead is in the hands of the laboratories. So it's important to test them regularly. And that could be, from our side, it could be a blind test to make sure that they test it correctly, but it's also important to require the accreditation and the ISO standards from our perspective. So thank you for listening to the current experience and how to use eDNA in a monitoring context, or how we think we can use it. And I hope really that this will lead to further debates and more knowledge sharing, so we don't go our separate ways when we put the eDNA to use. Thank you.